we start, whether you guys have any questions from yesterday, whether maybe you slept over things and something popped in your mind that was not clear. Okay, so if there's no questions, again, please feel free to interrupt me anytime. So we stopped here yesterday on this slide, and I wanted to talk with you a little bit about how we can actually measure variability at the sequence level. Because yesterday we talked more generally. Is this too loud, by the way? Or is it okay? Okay, fine. Yesterday we talked more generally about how we can measure heterozygosity or genetic variability. But now we want to apply what we talked about yesterday specifically to DNA sequence data and how we can sort of obtain quantitative measures of sequence variation. And so yesterday we also talked about this definition of what a locus is, plural loci. We said that this could be a longer sequence, or it could also be considered in some cases to be a single nucleotide position. So here we're actually thinking of a stretch of DNA in this context. So in studying DNA sequence variation, at least we often refer to a stretch of DNA from a region of the genome as a locus, even if only part of a gene was sequenced, or even if the sequenced region is non-coding. And then the independently sampled sequences we refer to as alleles. And so this is quite different from the terminology that we have used for allosine variability, where the locus was used to refer to a gene coding for a polypeptide. So here, a locus doesn't even need to be a gene, right? So I hope this is more or less clear. Right, so the basic information for quantifying DNA sequence variability comes from what we call haplotypes. So these are haploid stretches of the genome. Often as humans, we think of diploids. We ourselves are diploids. We have each gene in 2 copies. We have homologous genes, and we have pairs of chromosomes that are homologous to each other. So what we're considering here is only a haploid copy. So let's say an allele on one of our chromosomes, or we could also look at both copies of the hemoglobin gene from my genome, the one that I got from mom, the one that I got from dad. There might have been some recombination, but what we're considering here are haploid sequences. And so we refer to these sequences as haplotypes. So the above mentioned stretches of sequence from one of the 2 homologous chromosomes of an individual in the case of diploid genomes or organisms. Sometimes it can be a little bit hard from sequence information from diploid individuals to assemble haplotypes, but we won't go into that. Now, I just wanted to briefly show you how the data sort of look like. So the next slide shows an example of a sample of allelic DNA sequences for a particular gene, which is an enzyme which is called glucose 6-phosphate dehydrogenase, or G6PD. And so the example I'm going to show you is from this textbook by Brian and Deborah Charsworth. It's so they have a figure that shows sort of how the data look like for a worldwide sample of alleles at this locus in humans. The original data are from this paper in Genetics. And the interesting thing about this example is that for this locus, it's known that it's under selection because particular alleles can confer malaria resistance. So before I show you the slide, I have to apologize for the poor quality of my scan and for, it's really small, but I just wanted to make a couple of key points here. So first of all, we read this from left to right and from left to right. So from here, from left to right, and we would continue here. We would have the sequence. The position in the sequence is up here, and then we continue here. And what we have here vertically are, first of all, sample sizes here, 24, if I can read this, 4, 2, 2, and down here. So these are the sample sizes here in this column for these alleles. So 51 humans were studied here, or sequences obtained from 51 humans. Down here in the bottom row, you also have the sequence from a chimpanzee individual. Chimpanzees have the same gene, and we can look at the sequence in that region that chimpanzees have. And so then we have also these haplotype groups. You see here B, A minus, and A plus. These are different alleles at this locus, or different haplotypes. There's like the B1, the A minus one, and the A plus one. And it's written here in small print, the B alleles encode a protein with high enzyme activity. So you can think of the B haplotype as sort of being the normal version of this gene. And the really interesting one is the A minus one, which is here and here. So A minus, 7 people had this A minus allele in this sample, so sample size of 7 for this allele. And this A minus allele is a special allele because it's quite common, but it encodes an allele that has only 12% of the normal activity. We call this a deficiency allele because it has a defect. It's deficient in the activity of this enzyme, right? So the enzyme is working biochemically less well. But the interesting thing about this is that it confers malaria resistance to those people who have a normal B copy of the allele and 1 copy on the homologous chromosome with the A minus. So it's similar to the example that you might have heard about already in other contexts. There's an example that concerns the hemoglobin gene where people talk about sickle cell anemia, which is a genetic syndrome where some people have the sickle cell allele at the hemoglobin locus, and people who have 1 copy of the bad sickle cell allele and the normal hemoglobin allele, they're also malaria resistant. This is a very famous example of balancing selection that is in every population genetics textbook. This one here is simply another similar example. So, and then there's also the A plus allele, which is a mild deficiency allele that has 85% of the normal activity but does not confer malaria resistance. And then you simply have the nucleotides, right? The 4 letters of the genetic alphabet here along the sequence. So what's boxed here? The boxes here denote amino acid variants. And it's actually, so these dark boxes. So here is an amino acid variant. All the other sequences have a G here, but here in the box you have an A. This is an amino acid variant. It's an amino acid variant. It concerns an exon. Introns in genes are spliced out, right? They don't make it into the polypeptide. But so here we have an amino acid variant. And here, for also the A minus allele, there's a second amino acid variant at this position where the A minus allele has a G, but everyone else has an A. And so that distinguishes the A minus sequence or allele from the B haplotype, for example. So we can also ask, how is the A plus allele different from everyone else, from A minus and B? And the A plus differs from the other alleles by having another amino acid variant at this position, namely G. And then, this is really hard to see on this bad scan. With some fantasy, you can see that there's some boxes that are highlighted here, but without the bold outline. And so these are single nucleotide polymorphisms. So maybe we can look at this one. You see here, this is, well, maybe you can't see it, but this would be highlighted in the original figure. So there's a T here. 4 people had a B haplotype with a T, but everyone else who had a B allele had a C at this position. This would be a single nucleotide variant or a SNP, a single nucleotide polymorphism, okay? Now the question is, this is how our data would look like in this example. So how can we quantify how much variation there is among these different alleles and among these sequences that we have sampled? And so that's what we want to look at now. And so this, I'm going to use a hypothetical example that's going to be much simpler than what we had on the slide before. So how do we measure DNA sequence variability? And so here what I'm asking you to imagine is that we have a small sample. We have only sampled 3 haploid sequences. In other words, here, potentially at least, we have 3 different alleles. I mean, if the sequence is exactly the same, then we would have only 1 allele. Here it turns out we have 3 alleles. Each sequence is a little bit different. And we consider a very short stretch of 30 nucleotides. If I haven't messed this up, there's like 30 letters in each sequence. So here we have allele 1, allele 2, and allele 3. Now what we can see here, and which I have highlighted in red, is that we have a total, here in this example, of 4 nucleotide site variants or 4 polymorphisms, in other words, or 4 SNPs or SNPs. So the first one is here, because here, I mean, allele 3 and allele 1 have an A, but allele 2 has a G, right? So this is our first SNP or variant, okay? Or well, actually the second one, because like if we look here at allele 3, all these guys have a G, allele 1 has a G, allele 2 has a G, but allele 3 has an A, so that's another SNP, right? So that means we have this one, and then if we go here to allele 3, to these positions, we see that allele 3 also differs at 2 more positions in its allelic state. Here at this position, allele 3 has a C, but if I look at this correctly, we have an A here in allele 2 and allele 1, and here allele 3 has a T, but the other alleles have a C. So we have a total of 4 polymorphisms or 4 nucleotide site variants. And these are the data that we have. So how can we pack this into numbers to figure out how much variability there is? And to do that, we are going to use something, a summary statistic, that is called nucleotide diversity. Sometimes it's also called nucleotide heterozygosity. And the symbol that we often use for this is pi. And sometimes it's also called theta pi. I don't need to write it on the blackboard. It's fine. So pi or theta pi. So you will often see that symbol. And so what is this? For a given set of alleles sampled from a populations, pi is the frequency with which a randomly chosen pair of alleles differs at a given site. We already actually had that definition yesterday. It goes back to May 1973. So what we're doing, in other words, is we're going to make all pairwise comparisons that are possible, all unique pairwise comparisons between these sequences. That's in a way what this says. I mean, this is a little bit nonsensical here because I have only a sample of 3. What you have to imagine is a really big sample of sequences, and then, you know, probabilistically, you can like look at all pairwise combinations to see what's going on. And so that's what we're asked here to do. And so we can calculate this from data on a sample of homologous DNA sequences by determining simply the sum of the numbers of differences. So let's say I take sequence 1 and sequence 2 in my comparison, and I check how many differences do I have between sequence 1 and sequence 2. Then, for example, I compare sequence 2 to sequence 3, and I check again along the sequence, at how many positions do sequence 2 and sequence 3 differ? And I do that for all unique pairwise combinations. And then I sum these numbers up. So for the first comparison, I get like 3 differences. For the second comparison, comparing sequence 2 to sequence 3, I get 5 differences. I sum up these numbers, right? And then I need to divide that by the number of possible unique combinations that I've compared, and also the length of the sequence. In our example, the length of the sequence is 30 nucleotides. So this is just in words how we're going to do that. And what we need to do, as I've just said, is to divide this sum of the number of differences by the product of the number of sequences that were compared. So how many pairwise comparisons can we make? And so now maybe I'm going to go to the blackboard to illustrate this. So we have this formula that you might have seen before. I mean, if you have a physics or math background, you probably know this. As biologists, maybe not, but it's very simple. Simple combinatorics. So we have k times k minus 1, k minus 1 divided by 2. We can also write this sometimes in this form with this parenthesis, where I have the k up here and the 2 here. In words, this is K choose 2. I have K objects, and I'm asked to choose 2. But K might be much bigger than 2, so I'm asked to kind of like choose different combinations. So in our example, we had 3 sequences or 3 alleles, right? So I'm going to plug this into the formula. So K is 3, right? So let's say K equals 3. So this is going to be K, 3 times 3 minus 1 divided by 2. So that's 3 times 2 divided by 2 is 6 divided by 2 is 3. The answer, the magic answer is 3, right? And so how can we kind of like picture this? Because I find this very helpful to sort of unpack this little formula. Essentially, we have 3 items, and we want to combine these 3 items in all possible unique ways. So a good way of picturing this is like, let's call the 3 objects we have A, B, C, right? Now, how many ways are there to form pairs, right? If I have 3 items, there's 9 pairs here in this matrix, right? I mean, I'm sorry if this is completely trivial, but maybe for those for whom it isn't, I'm just going to write that down. So these are all 9 combinations I can do, right? But the thing here is, I mean, we're not going to get 9 as an answer, we're going to get 3. Why is that? Because first of all, we're not interested in the ones where we compare the item to itself, right? So these guys on the diagonal we can forget. So we have 9 objects, minus the one along the diagonal is 9 minus 3 is, I'm left with 6. But if we look above and below the diagonal, these are mirror images, right? So we're not going to care about those guys. If we care about those, we're not going to care about those or vice versa. So then that means we're left with 3 objects, right? And so that's exactly what we sort of need to do when we do these calculations. And then, okay, so we need to divide by that number, and then also the number of bases studied. Why do we also divide by the number of bases, like 30 nucleotides? It's because the number we're going to get, we want to express that per nucleotide. It's just like a convention. It makes things comparable and easier. It's like a way of standardizing the data, excuse me. So in our above example, we had K equals 3. We've just calculated on the black or green board rather, that this means 3 unique pairwise combinations. So, and now what we are asked to do is to count up the total number of pairwise differences between all 3 combinations of sequences. And here, that's going to give us 8, 1 plus 3 plus 4. But let's see where this comes from. So let's compare allele 1 to allele 2. How many differences between the 2? 1, right? Then we compare allele 1 to allele 3. We have 3 differences, right? And then finally, we compare allele 2 to allele 3, and then we have 1, 2, 3, 4. So that's 1 plus 3 plus 4 equals 8. To get the pairwise diversity now per nucleotide site, we divide this by 3 times, because k times k minus 1 divided by 2 is 3, times the number of sites. So 3 times 30 nucleotides. So 8 divided by 90 gives me 0.089. And so that's the probability that per site, the 2 randomly chosen sequences differ from each other. And this is our measure of nucleotide variability. And this is completely analogous to the heterozygosity or gene diversity age for the case of single nucleotides. And this is the measure that most people use. So when in my lab, or like anyone who is doing population genomics who's analyzing sequencing data, I mean, all these people are essentially calculating pi or zeta pi to figure out how much diversity there is. Now, the interesting thing, yes, please.
When you do that, I don't know, why you just not take all the values for each pair and you just have the mean or the distribution of that? Why do you, this measure is better, or why you choose?
But it is an average, right? It is an average because I'm averaging across all the nucleotide positions, right? That's why I'm also taking the 30 nucleotides into account. I'm even accounting for the nucleotide positions where I don't have a variant, right? Okay. They also count in my calculation. I'm sort of asking how many differences are there between two random sequences for the same gene on average. So it's an average measure. And so that also means in statistics or physics, we would maybe say it's an expectation in a way. Okay. It might not be true for every pair of alleles I'm going to grab, right? If I do this blindly, I have a huge sample, it's only going to be true on average, right? Okay. Yeah. Thanks. Now, this is pi, but there's another alternative way of doing these calculations, an alternative method. And this is based on the number of segregating sites, or capital S here, capital S with this subscript k, where k denotes the sample size, but often you will just see capital S in the literature. This is the number of segregating sites. What is the number of segregating sites in this example? The number of segregating sites here is also 4. 1, 2, 3, 4. These are just like where segregating variation means that we have polymorphisms. So it's just the number of polymorphisms. That's the number of segregating sites. And so I want you to think of this measure as the number of segregating sites. It's a little bit more complicated than that because we need to divide the number of segregating sites by the product of the number of bases, so 30 nucleotides, and the following sum, which is called Waterson's correction factor. So I didn't write it as a proper sum, but we can just put the summation sign, right? And I think it's the sum over 1 divided by I, where I is going to run from 1 till k, I think, if I'm not mistaken, right? K minus 1, thank you. K minus 1, exactly. So it's like this sum. I'm not going to explain where this correction factor comes from in detail, but we sort of need to take that into account. And what it accounts for is that not all the sequences that we're looking at are going to be necessarily genealogically independent from each other. So this thing has been derived by Mr. Waterson using coalescent theory, which is what you talked about with Sean Rice. But it's also, again, a simple measure, and in a way you can just think of it as the number of segregating sites, essentially. And so when we do this, we also obtain a per nucleotide diversity estimate, and this is now called not pi, but it's called zeta Waterson. The other measure was called pi or zeta pi. This is called zeta Waterson, or Waterson zeta. And this is also a very commonly used measure. So this is an alternative way of doing it. And so let's just, before I tell you about this line here on the slide, let's look at our example. We have 4 segregating sites. We have 3 alleles. Our correction factor, if we do the sum, we get 1 plus 0.5 equals 1.5. So let's see. So maybe I'm going to do that here, right? So we start the summation at 1. So what we're going to get is 1 plus 1/2. Right. So that's how we get this thing. And so what we're then doing is we're dividing the number of segregating sites by the number of base pairs in the sequence, which is 30 nucleotides times this correction factor 1.5. And the cool thing is that the number we get is exactly the same as pi, right? We got 0.089 here for pi, and here for zeta Waterson we get the same thing. This is not always true, but it's very often true. So if the population is actually at equilibrium, and if there's no selection, then we expect zeta Waterson and pi to have the same value. And so this also comes again from coalescent theory. So under so-called standard neutral conditions, standard neutral means a situation where the only game in town is genetic drift. There's no selection, no other evolutionary forces, just genetic drift. And the concept of standard neutrality means that it's neutral, the situation is neutral, drift is at play, but there's also no changes in demography. So we assume that the population sizes are constant. There wasn't any population bottleneck in the past, the population is not growing or expanding, population size is constant. That's what we refer to as standard neutrality. And under standard neutral conditions, these two measures are expected to be the same. If things are in what we call mutation drift equilibrium and in the absence of selection. Then we have two independent measures and they need to sort of agree with each other, zeta Watterson and pi. But under non-equilibrium conditions, which can also be neutral, this is, for example, when the population would shrink or grow, we might still be in a neutral situation without any selection. There might be deviations due to demographic forces like bottlenecks or population expansion, or non-equilibrium conditions could also be when selection is acting. The two measures will differ. And the reason I'm telling you this is because we can take sequence data, we can calculate pi, we can calculate zeta Watterson, and then we can compare the two. And if they don't match, then we know that we're not in a so-called standard neutral situation. It tells us that something else might be going on, and that something else could, for example, be selection. It doesn't prove that there's selection, because it could also be no selection, but a demographic change. Things could still be neutral, but there was a population bottleneck, or in fact the population is not constant, it's growing, and so that could also lead to a deviation, where pi does not agree with zeta Watterson. But if the two are not the same, under some conditions it might be a good first hint that what causes them to differ, these two numbers, might be selection, which we will look at in a subsequent lecture. And so also the two methods of estimating things differ a little bit, because Watterson's method only takes the number of SNPs into account, the number of segregating sites, which in our example was 4, divided by some correction factor. But pi is an average that also takes into account the frequency of the polymorphisms. It's not only the number of segregating sites, it's also how commonly they occur, okay? And so that's also why these numbers can sometimes differ. But if they are the same, it's a very good indication that things are standard neutral, right? So we often write this as, well, I think I have that on a slide, maybe on the next slide. Right, so under the neutral theory of evolution, variability in DNA sequences reflects the balance between the input of new variants by mutation, and of course these variants can then be lost again by genetic drift. And genetic drift obviously is due to finite population size. And so under this standard model of neutrality, the two measures of nucleotide variability are expected to be the same. So we can often write this here with this symbol E for expectation. Expectation is just the arithmetic average. It's actually a slightly strange word. So people in physics know the word expectation, I guess, no? I mean, that's more commonly used in physics than in biology. Essentially, it means the arithmetic average. But it's a little bit strange, right? Because if I have a distribution like this, the expected value here, the expected value is the mean, right? This is a symmetric distribution, the Gaussian distribution, right? And my expectation would be exactly the arithmetic mean. The same is true, though, if I have a bimodal distribution. But here my expectation would be here, and this is not what I expect, huh? Because here, I mean, so that's why the wording expectation is a little bit dumb. And you should be aware of this, because like what I'm expecting here is that I'm gonna, if I sample this distribution at random, I'm gonna find small people and big people, for example, right? So this is why the thing is a little bit misleading. But anyways, we use this symbol expectation, and under standard neutrality, expectation of pi equals the expectation of zeta Watterson. And so under this model, the variant frequencies at a locus are always shifting around, but we sort of reach a statistical equilibrium under the condition or assumption that the population size stays constant. And so both statistics, summary statistics, Pi and Zeta Watterson, are estimators, statistical estimators of this quantity that you talked about with John, and which I've mentioned yesterday, which we call theta, not Zeta Pi, not Zeta Watterson, but Zeta, which equals 4 times n, or NE, if we talk about the effective population size, times mu. I think, did you have a question?
Just want to know, when we are supposing that these distributions are normal, but the median is not a better way to measure that, because we would, the other day we discussed
yeah, so the question is whether it would not be better to use the median for
yes,
in some cases.
Because we were discussing
exactly. Yes. So usually when people use the simple expectation, what they mean is the arithmetic mean, as we have learned it in school. But more generally, there's things that we call measures of central tendency in statistics, and there's different ones. There's, for example, the mode. The mode is defined as the most common value in the data set. And this can or cannot be the same as the arithmetic average. For a symmetrical distribution, like the Gaussian or normal distribution, the mode is the same as the arithmetic mean, right? And would also be the same as the expected value. And then the third thing would be the median, which would of course also coincide here with the arithmetic mean. And so here, it's a bimodal distribution, so it's much better to look at the modes, right? And rather, the arithmetic mean is not going to tell us anything interesting, right? And for skewed distributions, like this, or like this, of course the median is much better. This, incidentally, you know about journal impact factors, no? You know, like the journal impact factor is the number of citations to articles in that journal averaged over 2 years. The journals that have the highest impact factors are, for example, Nature and Science and things like Neuron, like every scientist's dream, or many scientists dream to publish in these papers. Because like they have such a high impact. It means if you get a paper there, it will have a very high likelihood of being cited and read by everyone. But it's an average, this measure, this impact factor that Nature and Science are so proud of. It's an average, it's an arithmetic average, which just goes to show that the people who have invented the impact factor are really bad statisticians, because the distribution of citation impact is something like that, right? I mean, it's highly skewed. Most papers in Nature and Science get only cited very few times, as similar to other journals, and only very few papers in Nature and Science get a lot of citations. And in such circumstances, the median is a much better measure than the arithmetic average, right? And so that's why journal impact factors are stupid. Just as a side note. Right. So, but let me just briefly say something about this theta that you looked at with John. I don't know, do you guys remember a little bit where that comes from? I think you derived it using coalescent theory, more or less. Okay. So it's like a theoretically derived measure. And so more less than more. Okay. So I'm sure you looked at some of these coalescent trees or genealogies, right? So here, this is the present time. Present. And up here is the past. Present, past. And what we're looking at here is a sample of 2 allele copies. Think of in our example with the glucose 6 dehydrogenate enzyme, think of like 2 sequences from 2 individuals that we have in our sample. And what we're asking in using coalescent series, how they're related and when they had a common ancestor, a most recent common ancestor, MRCA. Because in fact, I mean, these two copies of these two alleles, they could go back to the same individual, right? In principle. And so this could also be extended to more, of course, than 2 allele copies or samples, but like the easiest is to imagine 2 for simplicity. And so often what we do in these coalescent models is we have such a sort of genealogical tree, and we're looking back in time from the present. So this is our sample today, and we're trying to infer something about the past. And what we're imagining here is like these 2 allele copies at some point in the past, they must have come from the same individual. And we want to figure out when that happened. And then in these processes, so how, for example, if these 2 allele copies differ, it's like 2 sequences. They can be related, but they could differ at 1 or more nucleotide sites. Where can differences come from between them due to mutation? And on this tree, where can mutations happen? Mutations are very rare, so they could like happen on this branch, or they could happen on this branch. In this model, we're supposed to assume that mutations don't happen simultaneously on both branches and at the same position. And this is reasonable to assume because mutations are sufficiently rare, right? But so we can just like sprinkle mutations on this tree. And so with John, I'm sure you talked about the expected coalescent time. So here, from the present to the past, we can actually figure out the average time for these 2 guys to coalesce to the most recent common ancestor. It's an expectation, it's again an average, and the answer is 2n. This is the expected coalescent time per generation for a pair of alleles, or for 2 gene copies. It's 2n. Again, it's an expectation. There's a variance around this. It's not always 2n. But this means it takes, on average, for 2 allele copies to converge to their common ancestor, on average, it takes 2n generations, right? It's not years or months or whatever, it's generations. By multiplying this by generation time, we can figure out the time scale. And so, in a way, this is a good picture to figure out, qualitatively speaking, where the 4n mu comes from. Because zeta is 4n mu. And the different way of writing this is simply 2 times 2n times mu. Mu is the mutation rate, or the probability that a mutation happens. 2n actually is a time here in this context. Of course, at the same time, it's the number of allele copies. But here in this context, it's an expected time. It's the time to go from the present, where I have these 2 guys, to their most recent common ancestor. On average, this is 2n. So this is a time, t. And here I have the mutation probability. Why 2? 2 branches, exactly. The mutations can happen either here or here. Or another way of thinking about this, if 2n is a time, let's call it t, how much total time do I have in this tree? Total time. 2 times 2n, right? And so why do I need to take the total time into account? This is related to the total length of the tree. 2n is the height of the tree. The total length of the tree is 2n and 2n. That's the total time. The total length of the tree is important. Why? Because per unit time along the tree, I can have mutations. And the number of mutations I'm going to get is proportional to the total length of the tree. Does that make sense? Yeah, okay. So that's very intuitive how you can think about where the 4n mu comes from, which looks so mysterious, right? It's not so hard to understand. You can think of this as a total time times the mutation rate. And this is going to be proportional to the differences we're going to see between 2 sequences. In other words, this should like give us in many situations, pi should give us this result. Or theta Watterson should give us this result. So this is just a little detour into this. So now let's look at some empirical estimates of how these numbers look like in the real world. So these things have now been measured for practically any organism that we can conceive of by sampling sets of homologous genes from natural populations and sequencing them. And so these patterns of variability that can be quite different among different organisms. So here, I mean, these are rough average values over many genes for SNPs that do not affect protein sequences. But for example, if we look at E. coli, we have like 0.05. Drosophila here, it depends a little bit where they're from. This is actually a quite high value. It's 0.02. Homo sapiens is like 1 in 1,000. What does this mean, 0.02? It means that, so this is 2 in 100, right? 0.02. It means that if I have a stretch of DNA that's 100 base pairs long, on average, I will find 2 positions that are variable on average in a longer region. And here in humans, you see that this is an order of magnitude smaller. So humans are less variable than fruit flies. 1 SNP position in a sequence length of 1,000 base pairs. So a value of zeta Watterson or pi of 0.01, 1% implies that there is 1 SNP every 100 base pairs. So that's like sort of what it means. So if we take account of the genome size, these values imply, of course, that there can be several millions of SNPs. People always think like, mutation rates are so low. Mutation rates might be 10 to the minus 8, and genetic variability might be very low. But like, you need to compound that with the fact that our genome is really long, right? I mean, I think the length of the human genome is, I think, if I'm not mistaken, 3.2 times 10 to the 9 nucleotide positions. I think so. It's huge. And so even if you don't have that often a SNP occurring, you count up the numbers, you have a huge reservoir of genetic variability. So here, this is again from the Charlesworth textbook. Here are these numbers in percent plotted. You see humans are not very variable. But then this thing here, Siona intestinalis, is super variable. Sea urchins are also not doing bad, or this nematode worm. So you can see then, Plasmodium is apparently not very variable. Then something like Salmonella is super variable. So this depends on the organisms. Why does it depend on the organism? Generation time. Mutation rate, I would say. Because the mutation rates can be quite different between organisms, right? That's one thing. And also the N, by which I don't mean the time now, but the number of individuals. More commonly, we think of this not as N, but as NE. The effective population size can also be quite different. So that's why these numbers can be quite different depending on the organism. Yes, please.
But there is a reason for the mutation rates to be so different between
Yes, that's
species?
interesting. I mean, we're not going to have much time to talk in detail about this, and I'm also not an expert on this, but we know in evolutionary genetics that mutation rates can evolve. And sometimes they can really change during the time course of evolution. And so, for example, this is a relatively trivial example. In some bacteria, you sort of have a normal mutation rate, but sometimes you get genotypes that are called mutators, and they have a much higher mutation rate. Now, the interesting thing is that it's not good, right? If mutations are too common, that's usually really bad. But if they're too infrequent, you don't get any genetic variability. And so most mutation rates are sort of like on a sweet spot. Mutations are rare enough not to cause too many issues, but sort of frequent enough so that you have, in a way, enough genetic variability. That sounds very teleological. I don't mean it this way. But mutation rates can indeed evolve. And you can easily see that if copying errors, if things are quite well adapted or quite well functional on average, then having too many copying errors would like really be bad, right? I mean, you could imagine that there could be selection for having too high mutation rates. Of course, then sometimes it could be good if you can change faster. But then we also need to keep in mind that most changes are practically always or almost always deleterious. Yeah. So let's look at the brief example. So here, let's replace n by the effective population size that you have talked about with John, I'm sure. Yes, please.
I just want to ask, if we calculate this value of mutation rate and everything, we can, if we have, I don't know, ancient DNA, we can try to figure out in what moment of the history of this bacteria or organism specific gene compare, or maybe an estimate? I don't know.
Exactly.
Because here is just selection, without selection, right? So
yeah, exactly. So we can date things. We can try to figure out, how long, we can look at the particular gene and the alleles in a human sample of sequences, and we can sort of figure out when in the past did this thing originate, exactly. And so I think that's what I'm gonna try and illustrate here. So let's say we know the mutation rate, and nowadays we can sort of measure this quite well, or estimate it quite well. And so let's assume, so this allows us from this simple formula, so we know that zeta Watterson or pi equals 4 NE mu, right? And so it's really muggy, right? So we can actually solve this little formula for NE, for example, the effective population size, if we know mu. And pi, let's say, we can just figure out from sequencing. We can just like do the simple stats that we've done on the slides before. So for example, we know in Drosophila, let's say the mutation rate is 4 times 10 to the minus 9. And let's say we've measured the pi value empirically from sequencing data for an African sample of fruit flies of 0.02. And if we solve this little equation for NE, what we will get is an effective population size of 1.25 million flies. This is the effective population size. This is not the census size, and I want to stress this. It's a little bit hard to picture, conceptualize what NE means. I'm sure you have discussed this. I'm going to say something that is wrong, but still maybe vaguely helpful. I mean, a purist would like probably kick my butt for saying this, but like, I mean, so one way of thinking about it, one way of perhaps thinking about it is that NE, you can think of how many individuals actually contribute to the future generation by producing offspring. So the number of breeding individuals. This is not exactly correct, but I still find it quite useful. Because like, I mean, so we're going to talk about humans in a minute, but in humans, population size is what? 8 billion? 8 or 9 times 10 to the 9? And it's obvious that not all these people are going to contribute children to the next generation. Not, if you think about genotypes, not all these genotypes or lineages are going to contribute to the next generation. And so I think it's somewhat helpful to think of NE this way, even though it's not entirely accurate. I'm happy to talk about this more in a break. But anyways, let's do the same thing for humans. So let's see in humans, and let's say that we find that pi is 10 to the minus 3. And we know from several studies that the mutation rate is typically on the order of 10 to the minus 8. And so we can do this calculation again. What will we find for humans? 25,000, right? We're like 8 or 9 billion people on this planet. I'm not sure that's such a great thing, but there we are. But the effective population size is 25,000. So if we think about this as individuals, that's not very many individuals. What is the meaning of this? What is the meaning of 25,000 people? So maybe, do we have something to erase? Yes, it's here. So NE is 25,000. We know that the mean coalescent time is 2 times N or NE. That's the average coalescent time. That would be 2 times this number, right? which is 50,000. So that's the mean coalescent time. If we think about a pair of allele copies we have sampled here for a given gene, would be 50,000 generations. Generations in my bad handwriting. Now, how many years does this correspond to? A lot. Exactly. So often for humans we assume the generation time is 25 years. It's not entirely correct necessarily, but 25 years. Some of you are probably 25, right? So, and we only, I'm sorry, but we only think about the girls here, because the way generation time is defined is it's the average number of the average mother when she has her first child. I know that, like, I mean, of course, there's like women now who have children much earlier than 25, and nowadays it's also very common to not have children or have them very late. But as sort of a, kind of like, you know, a rough number, for humans, we like to use a generation time of 25 years. Whatever that means or not, but we're just gonna multiply this 50,000 generations by 25 years, and then we get a big number, as your colleague said, namely 1,250,000 years. So this is the mean coalescent time using that effective population size, using the mutation rate and the sequence variability we have measured. This is really interesting because, like, how old are modern humans? Do you guys know? 200. Who said 200? 200,000. Modern Homo sapiens, 200,000. How can it be? I mean, so I look at two hemoglobin alleles. It could even be from the same individual, we can do this. But let's say we take two random alleles at the hemoglobin locus. We do this mumbo-jumbo calculation business, and we figure out that the coalescent time is like deeper back in history than the origin of modern humans. It means that before we became modern humans, we were something, right? We were not created ex nihilo. We inherited something from our ancestors that was already there. Genetic variability that is already there. What we're doing with coalescence and with these calculations and considering this effective population size is we're looking back in time. This is really cool, like especially if you're a physicist, right? With a telescope, you can look back in time. With a particle accelerator, you can look back in time. With coalescent serine population genetics, we can look back in time. We're looking back in a time when the human population was much smaller and mutations are super rare, right? And so that means that, like, most of the genetic variants that we have are quite old. And many of them we might even have inherited from our very distant ancestors, even before we split off in the genealogical tree to become modern humans. And there's lots of polymorphisms that we share with chimpanzees and other primates that go back to very, very deep back in time in a way. So this is sort of what I wanted to say about this thing with exactly effective population size and looking back in time. So this is like one way of thinking about these things. Most variability is old, and we're looking back in time. We're not really caring in population genetics about the fact that there's 8 or 9 billion people. I mean, nothing against that, but it's just like not relevant for what we're doing here. Someone, I think, asked me yesterday, could this not matter in the future? I mean, if we would like do this thing in a bunch of 1,000 years or in 200,000 years, maybe the current population size would become more relevant. But here, we're measuring something now, and we're looking back in time, and we are coming up with these numbers, right? So let's see where we are. I think we're actually going to skip over this. Are there any questions? So the whole purpose of this sort of lecture was to introduce you guys to pi and zeta Watterson, because we're going to use that when we want to detect selection. So I have a bunch of more slides, but I'm not sure I want to go through them. So let's see what we could do.
Can I,
Yes,
just to be
please.
sure, this is not considering selection, right?
Yes.
Okay.
Yes. Yeah. So exactly. So this is not considering selection. Yeah.
And
Because selection could speed up things. Yeah.
and this year is like 1,002,000.
Yes.
And
It's years.
Years in to the most
Recent common ancestor.
recent common ancestor
Yeah.
of all humans.
Well, yes. If we believe these numbers that we put in, yes. I mean, so this is like true on average, considering this estimate of pi and this mutation rate. Now, the mutation rate is not always 10 to the minus 8 along the human genome, but it's a quite good number. So, you know, these numbers could vary a little bit,
And how
depending on where in the genome you would look. So maybe just as an example, so in Drosophila, we've seen that the effective population size is on the order of 1 million. And depending on how you estimate things, there can be some variation. I'm trying to say that we shouldn't believe too much in the precision of these numbers, but roughly speaking, I think it's sort of what we would get. Not for everything, but yeah.
And how do we get into the 200,000?
Mm-hmm.
It's this the number? I think it's 200,000
For modern
years
humans,
for
I think
modern
that's
humans.
Yeah.
How do we get there?
Well, it's like
with, I don't know, some analysis of carbon or something.
Well, I mean, we have fossils.
Yes.
And then people did probably radiocarbon dating. And nowadays with sequences, we can also look at things. So this is actually really cool. I mean, like, this is not my field, but I have a colleague in Fribourg, Daniel Wegmann, who is working on ancient DNA, and he's doing population genetics on humans. And as you know, Svante Pääbo just won the Nobel Prize for doing all this stuff. Like, he sort of founded the field of paleogenomics. And so this really enabled, which first was a technological challenge, to reconstruct the genome of a Neanderthal, which is amazing. I mean, first they did it from mitochondrial DNA, and then they managed to do it from nuclear DNA. And you can do all these things. And so I think when was it? Neanderthals and humans split off like something like 800,000 years ago? I don't remember the numbers. I'm not an expert in this area, but like, you can figure out how much of the genetic variability is shared. How different are Homo sapiens from Neanderthals? We apparently overlapped. We interbred. And something like 2 or 3 or 4% of our genome on average are Neanderthal sequence. There was like a recent paper saying that like people who suffer from long COVID, apparently, I'm not sure how solid these data are, but there was a paper, I think, in Nature, showing that like the parts of our genome that make us more prone to developing long COVID are Neanderthal-related sequences, things like that, right? And so it's pretty amazing what you can do. And also with this sort of coalescent approach, looking back in the past. And, yeah. Yes, please. So this is much better than me showing slides. If we chat a little bit, it's good.
I was just checking the years, and what it appears is that the Australopithecus robustus is like that number. The Australopithecus robustus in the millions of years ago. So
yeah,
fun fact.
Good. So let's see. So I think we still have half an hour, right? Do you guys want? Yes, please.
Okay, I'm not sure if I understood the effective population size, because look, I don't know, it's like the population.
What it means, really?
Yeah, it's the population that contributes genetically to the next generations, or something like this, because, you know.
Again, a purist would not be happy with me. Let me try to explain what it means maybe a little bit more technically. have you guys talked about the Wright-Fisher model? With Ricardo, I know you have talked about the Moran model, but I think that's a long time ago, right? Isn't it? Right. Okay, so the Wright-Fisher model, W-F, Wright-Fisher, because I'm too lazy to write, or the Moran model. These are simple probabilistic statistical toy models. Actually, they're models of what some of you in physics or statistics might know as Markov chains, where you have transition probabilities. You go from one state to the next with some probability. So let's say state 1 to state 2. So this would be sort of a Markov chain. Here we have transition probabilities. Markov chains have the property that they're memoryless. So this state here just depends probabilistically on what happened, on the state before, but not what happened in the past. And so if we think about populations of alleles, if we have random genetic drift are random fluctuations. The way we think about random fluctuations is doing random sampling, right? So I often like to think about an urn or a bowl, with different balls, which are my alleles. Right, so here I have like two alleles, the filled balls and the empty ones. These are like, let's imagine one gene with two alleles, and these are my individuals. And so what we're doing with these models is kind of like random sampling of these balls. We'll put them back into the ball, but we look at the ball, and depending on the ball that I pick out here randomly, I'm gonna, propagate my next generation. That's what we're sort of doing. I mean, in the Moran model, it's a little bit a different way of doing it from the Wright-Fisher model, but it's essentially the same thing. So we're doing random sampling, and via random sampling, we're building the next generation of individuals that have different allelic states. And then I have that generation. Then I do again random sampling to build the next generation, okay? And so these models assume that the population size is constant. That's just a convenience so that we can do these things simply. So for example, I don't know. I'm just going to draw this here badly, but I will try. So for example, in the Wright-Fisher model, I would like have that many individual alleles for the same gene. This is generation T. This is generation T plus 1. So I'm now looking forward in time rather than backward in time. This is like sort of the coalescent upside down. And so here I have 2n allele copies. And in the next generation, my task is to again have 2n of these guys, right? And so what I'm doing is like, I'm randomly sampling. So let's say I go into my urn and I call this individual here, for narcissistic reasons, I'm going to call this Thomas. That's me. And this is my, so let's say I make two types of sperm. I make this sort of sperm and this sort of sperm. These are the alleles. These are like haploid gametes. So it can be that two of my children, incidentally, I really have twins, that if I go in that urn, I could do it blindly and I pick out, I look at the thing, Thomas. Then I put it back. I do the same thing again. I was like, Thomas. The very same Thomas sperm that has contributed here two offspring individuals, right? And so we kind of like play this random business. And so this is like what we sort of do in the Wright-Fisher model. And this guy here, I don't know, we shouldn't give him or her a name, was unlucky and did not leave, maybe did not contribute offspring to the next generation. And so the constraint is that we always need to keep this as 2n. So it's like a simple random sampling scheme. And the same is true for Wright-Fisher and the Moran model. They just do it slightly differently. And this is our model of the evolutionary process where we have genetic drift, because the only thing that happens here is genetic drift. There's no selection, no mutation, no migration, nothing. So it's a model of genetic drift. And the coalescent theory is another way of looking at the very same thing. And from the Wright-Fisher model or the Moran model or from coalescent theory, we can derive all these things. Like, for example, for n mu. So we can calculate these things. So this would like be the expected genetic variability that we can calculate from these sort of simple toy models. Now, what is NE? So let's say you go into a real-world population and you measure sequence variability, genetic variability. And maybe the number of individuals you have looked at could be some number, I don't know, 500 individuals or so. But, and so they correspond to some level of genetic variability pi, let's say. That's what you have empirically measured, and you know it's from that many individuals. But in your Wright-Fisher model, or in your Moran model, that very same number pi might come about by a much smaller number of individuals. And so this would be NE. NE is the number of individuals that gives you the same amount of genetic variability than the population you have really observed. Often that NE is smaller than the number of individuals you have sampled. Again, this could be, so one example is in seals. In seals, males have harems. And so you can measure a bunch of seals and genetic variability in the male seals and in the female seals, and you have a certain sample size. And what you will see is like the census size does not really matter in a way. What really limits genetic variability is the males. So let's say, I don't know, maybe your sample size is going to be 500, but there's only, let's say, I don't know, 20 males and 480 females, because like each of these males has many females. These guys are in a way like a genetic bottleneck. In the next generation, when I measure genetic variability, all the genetic variability that was present in the generation before had to pass through these 20 breeding individuals, right? So this number here is completely irrelevant. What is relevant is NE. And so I can take a Wright-Fisher model and I could play around with the numbers. I don't know what this situation corresponds to, and see what amount of genetic variability will be matching the one that I really observe. And that will be the corresponding effective population size. And in most cases, it's going to be much smaller than the number of individuals I count. So it's sort of a theoretical calculational device. There's nothing in the real world that is really the effective population size. But one way of thinking about it loosely is maybe the number of breeding individuals that contribute to the next generation, even though this is not completely true. So this is more or less what it means. If you look it up on Wikipedia, it will say something like that. The number of individuals in a Wright-Fisher model that gives you the same amount of genetic variation that you have observed in your real-world sample.
Okay, thanks. But then I have another question that is, when you know when to believe in your model, because like,
yes,
if I'm doing this for the humanity and then I see this number of effective population, I would just think that the model is wrong somehow, you know. So when to have doubt about the model and when to believe, even when you have this type of result that is kind of counterintuitive.
Yes. I mean, so first of all, we have different ways of modeling things that are sort of in the limit equivalent, like the Moran model and the Wright-Fisher model. In many situations, or the standard coalescent model, they will produce the same result. So these models, that suggests that these models seem to be somewhat robust. They do make simplifying assumptions, right? For example, the assumption that like the population size is always constant, things like that. But with the effective population size, we can account for this. With the effective population size, I can calculate an effective population size for a situation where the population size is fluctuating over time in the real world, or where the sex ratios are not equal, where there's not the same number of breeding males and females. So in Hardy-Weinberg, which is the simplest model, we assume all these things. We assume random mating, the population size is constant, doesn't change, same number of males and females. These are like all these idealistic assumptions. And the effective population size actually sort of allows us to relax that and still make sense of the data. And then, you know, so I mean, of course, there's uncertainty sometimes in these estimates, but I would say this gives us already a pretty good idea of what's going on. I'm not sure this answers the question. I mean, these are not really precise numbers, I would say, but they give us some idea. I think of them as numbers, but I think of them often as qualitative numbers, because in the sense that like it gives you a ballpark figure, right? Like physicists are into approximations and not into pure math. It's like a ballpark figure. Who cares, is the effective population size of Drosophila 1 million or 1.5 million? It gives us an idea about how much genetic variability there is or was in the past. Yeah. Cool. So I think this is soon going to be it. So let me just see what we want to do next. Okay, maybe we can look at a few more things. I'm going to skip over some things. Where does all the variability come from? Obviously from mutation. So this is not what I want. So mutation is the ultimate source of all genetic variation. And so we can now measure mutation rates quite accurately. Actually, this estimate of 1.2 times 10 to the minus 8 mutation rate for humans comes from this Nature paper here in 2012. And so, as we've discussed, mutation rates can be a little bit different in different organisms. We know that, like, you know, in some RNA viruses, they can be quite high. And in humans, they're like on this order, 10 to the minus 8, 10 to the minus 9 in Drosophila and in some bacteria. And what this probably means that mutation is not going to be very effective in terms of changing the composition of population. Too rare, right? And so usually, even though to do these calculations, we need to know the mutation rate, in a way we don't need to care too much about mutation. I mean, mutation is not a really major directional force in evolution. It's in a way much less important than drift and selection, right? And so I just wanted to briefly show you a simple model of how we can think about this. And I'm not sure you guys have done this, probably not. Did you talk about how mutation can affect allele frequencies with John, or no? Okay. Okay. So consider we have two alleles, A1 and A2, at a particular nucleotide site. And we assume here discrete generations, but that's sort of besides the point. So let's say that if I'm an A1 allele, I can mutate with some low probability into A2 at a rate U, and A2, the A2 allele can mutate back to A1 at rate V, right? So maybe I'm just going to draw this again. So we have two alleles, and they can change state by mutating. So A1 can mutate into A2 at rate U, and A2 can mutate into the A1 type at rate V. So that's what is written here. So now let P be the frequency of A1 and 1 minus P or Q the frequency of A2. If the population is so large that we can ignore random changes in frequency, then the frequency of A1 in the next generation is going to be P prime. And what is this going to be given by? So here I have the frequency of the A1 allele in the previous generation. And what is mu? Or U. U is the probability or the rate at which A1 changes into A2, but I don't want that. So 1 minus U is the probability it's going to stay an A1 allele, right? Times the frequency. And so with this probability, we're going to get a contribution to the A1 alleles in the next generation, right? These guys are going to contribute to this pool here of A1 alleles in the next generation. But then I also have A2 alleles, right? A2 alleles have a frequency of 1 minus P or Q. And why do I care about A2 alleles? I care about them because they can mutate from A2 into A1, right? So I get this expression here. And if we do the algebra, we can calculate the change in frequency delta P by subtracting this stuff here, P prime minus P gives me delta P. And so what I'm left with is like a simple expression, which is a function of the two allele frequencies and the two mutation rates. And what this means is that the rate of change in allele frequencies due to mutation pressure is on the same order of magnitude as the mutation rate. That means mutations could change allele frequencies, but mutations are very rare. So the time scale on which the process of mutation could appreciably change allele frequencies is the time scale of mutation rates. So if my mutation rate is 10 to the minus 8, that's the probability I have a mutation. What's the time scale? It's the inverse of that, 10 to the 8 generations roughly, for example. So this means it's incredibly difficult for mutations to change allele frequency. So when we consider allele frequencies, we don't need to care about mutation. We need to care about mutations because they're the substrate or give us the substrate of genetic variation. But if we think about how allele frequencies change over time, we can sort of safely ignore mutations. This is the point I was trying to make here. We can actually calculate the equilibrium frequency here. So when delta P equals 0 means the allele frequencies are not changing anymore. Under this simple model where the only game in town is mutation, I can calculate the equilibrium allele frequency, and it could be whatever, it could be 0.5, but like it takes a long time to get there, impossibly long, and before then other things will have happened. So drift is a very powerful way of changing allele frequencies, selection as well, but mutation is not. So this is what we have here. So this means that evolutionary forces other than mutations, such as selection, genetic drift, which act more quickly, are likely to be much more important in controlling the frequencies of the variants that control to variability. And the other point I wanted to mention here is, where does the variability come from? It's mutation. And what about selection and drift? Selection and drift are forces where variability gets lost. Mutation creates variability, but under selection and drift, variability disappears, unless there's some forces that will maintain it. One exception for selection is balancing selection. But other forms of selection, purifying selection, positive selection, they all lead to a loss of variability. So we have a constant loss of variability. This is counteracted a little bit by mutation. So therefore, we have things like mutation drift equilibrium, mutation selection balance, or mutation selection equilibrium, and these things that maintain variability. But yeah, mutation does not really change allele frequencies. Okay, so it's 10:02, and I think I'm gonna skip over this stuff, and then maybe just finish by briefly talking about FST. So all this stuff that we're talking about is really setting the stage for understanding selection. And so what I wanted to end up with, so now we talked a lot about genetic variability, but there's also a concept that is called genetic differentiation. And what we can ask here is, for example, we can imagine two populations and we can ask how genetically different or similar are they from each other. And this can also be useful for looking at selection. So we have seen that, for example, in previous classes you have heard this, that genetic drift alone can be enough for two populations to diverge. Just by chance, they become different. So they start out the same, with the same allele frequencies at the loci we consider, but you let them go independently. And what can happen after a long enough time, one population might have become fixed for one allele by chance, and the other population might have become fixed for the alternative allele. And so this is genetic differentiation or divergence simply due to drift alone. But it's also clear that selection could also lead to divergence or differentiation between populations. Selection might favor one allele in that habitat, but another allele in another habitat. And so genetic drift can lead to this, and selection, migration, as well as spatial distance can also contribute to divergence or differentiation. And so one way this can be measured is by this statistic here that is called FST. I'm not sure, did you talk about this in previous classes? Not really? It's just like a measure of how genetically different things are. And we can calculate this from genetic data. And I don't want to go into detail here, but we can just like sort of look at this way. So it's a measure of among population differentiation. And it sort of is a relative measure, or a relative proportion of the genetic variation that exists among subpopulations as compared to the total pool of variation. So here I have two subpopulations and they sort of form a meta-population. And then comparing the variability in these two subpopulations to the total variability, right? And so FST is sort of a summary statistic that measures how similar or different allele frequencies are. So for example, and it can go from 0 to 1. And if FST is 1, it means maximal differentiation. It means, so for example, if I look at the single locus with two alleles, one situation could be that in this subpopulation, every individual is fixed for everyone has the capital A allele, but in this subpopulation, everyone has the little a allele. Could be not more different, right? And this would be FST equals 1. But if all these guys share the same allele, then there would be no genetic differentiation, the FST would be 0. And so this is a measure of how genetically different things are that we can use for sequencing data. And the reason that I wanted to briefly tell you about this is that sometimes when we scan in the genome, we go along the genome and we look at the sequence, we often calculate FST. We compare, for example, the sequences from two populations. And we try to figure out, are they different due to selection, for example. We're often calculating FST along the sequence to see how different the samples or the populations are. And if they're super, super different, and we can sort of rule out other things, it might mean, possibly, that they're different due to selection, okay? It doesn't prove it, but in some situations, it's quite likely. And so people do what are called FST outlier scans. They look for unusual patterns of FST. Now, maybe just as a brief prelude that we're going to talk about, it could be that you have big differences in FST between populations, for example, because at the beginning they were the same, but they had the same genetic variability, but they have been living apart for a long time. And maybe they're spatially isolated from each other. They're not exchanging any migrants anymore. Or they're isolated by spatial distance. We call this isolation by distance. So even if you start with everyone being more or less the same, given long enough time, given long enough distance, these populations will have a life of their own. And they will end up at different places, genetically speaking. They might end up like this. And there would be no selection, right? But this is demography, this isolation by distance. This should be true for many loci in the genome, okay? If I see a genome-wide pattern of isolation by distance with very strong FST differentiation between populations, especially at loci in the genome where we think they're neutral, then I'm led to believe that this difference between these two populations has to do with demography, not with selection. What is special about selection is that, yes, selection can happen anywhere in our genome, but selection is still sort of rare. Beneficial alleles or mutations are rare. So selection, if I look at the genome, should leave a much more localized signature in the genome. Whereas demography, like things like genetic drift, isolation by distance, spatial structure, should maybe leave a trace in the genome at many nucleotide positions for many genes. But selection might be much more specific and localized. So if I see a FST outlier that is very localized in the genome, then maybe this is more compatible with selection than with demography. Because if it's just isolation by distance, I should see this at other loci too that have nothing to do with selection. So this is sort of the idea. And I think next time we will probably talk about how we can use this measure and other measures, and also how we can use pi and zeta waters to sort of get some idea about whether selection has been acting. Right.
Thomas?
Yes.
Hi.
Sorry.
I was going to say professor, but then you
No, say Thomas, please. That's good. You get the full grade.
I thought I would be reprimanded. Can you expand on this about selection being when we see a very restricted signal in one loci, in one locus, and the other forces being when you see that spread? Because when I think of like, if you have a protein, I can imagine it being selected on basis of one single amino acid, right. because then this activity, the activity the enzyme does will be better or the like. But for very complex traces, I think you should probably have simultaneous and various and simultaneous changes. So, to exhibit this phenotype which is being selected, you need changes in this protein and this protein and that.
I think it's completely true what you're saying. I mean, I was oversimplifying a little bit. So the way I think about this is often, many of these things that you're describing probably happen sequentially, not all at the same time necessarily. That you need to have this mutation, that mutation, maybe a mutation in that gene to make a complex adaptation, like a polygenic adaptation that involves many loci. Usually the way I think about this, that like all these good beneficial variants that we needed at these different loci to evolve this complex phenotype, they probably didn't all arise at the same time, right? Of course, it's not impossible that a lucky individual had all the right mutations in these genes to sort of have that phenotype, but I often think about this sequentially, and then I would argue that my picture still holds. I mean, so if we quantify genetic variation and we look at what is called the distribution of fitness effects, which is often sort of a gamma distribution, we often see that actually the biggest class of de novo mutations that happen anew are deleterious, they're bad. Every mutation is bad. Almost every mutation is bad. Many of them are slightly deleterious, some of them are really lethal. And then we also have a rather big chunk of mutations that are neutral or nearly neutral. But if we look at that distribution, the smallest part of that distribution, a small tail, are really the beneficial mutations. What this means is that beneficial mutations are rare. So I agree with you, but still, if we look at new mutations, most of the good ones will be very rare. And that also means that, like, if we look in the genome, it's quite unlikely that we will see a lot of selection signal, right? Whereas, for example, think about the demographic event. Think about the population bottleneck. We go to some place, and then the poachers kill many, many individuals and only a few cheetahs survive. A huge genetic bottleneck. If we look at the genomes of these cheetahs, many genes in the genome will show that reduction in variability, right? So we see the bottleneck, this demographic event, genome-wide. But with selection, we would probably see a more localized signal. Not always, but often. So the sort of intuitive argument is that selection, in a way, is more rare than genetic drift or a deleterious mutation happening. And the other slogan is that demography tends to affect the whole genome, but selection doesn't. Selection never sort of has a pangenomic effect, but demography, like especially a bottleneck, probably would have. Yeah. And many more sites in the genome that are expected to be neutral or deleterious than under positive selection. And so that's sort of the argument when people say, there's like an amazing localized FST outlier, but everything surrounding in the sequence does not show this strong statistical outlier signal. This is sort of the logic that people use. It's not a proof, obviously. Yeah. Does that? Yeah.
It does. I think it's just the trouble of trying to distinguish what is chance by itself and what is a cause and effect relation.
Yes, I think that's a very good point. Actually, we will talk about this more because I hopefully towards the end of the week I can show you some examples of my own work where we can like do all that stuff. But ultimately, I would argue that if we want to be sure about selection and adaptation, we need to do some experiments to really show, right? I mean, the Higgs boson, for the physicists, the Higgs boson is beautiful as a theory, right? By a bunch of people, including Peter Higgs. But it's useless unless you can really demonstrate in a particle accelerator that it exists and complete the standard model. I would argue in population genetics, the theory is beautiful in the statistics, but at the end of the day, we sort of need to do experiments to show that the thing is real. Yeah. Okay, great. So I think we're already 2 minutes past the hour. And well, thank you. And see you guys tomorrow, I guess.